This video goes over the sample genetics problems for alternative patterns of heredity. Through these problems, you can compare and contrast Mendel's rule of dominance to other patterns of heredity. We'll examine how incomplete dominance compares to complete dominance. Note that the flowers on the left-hand side are Mendel's peas, and on the right are Snapdragons. Let's take a look at Mendel's peas, and this is going to show complete dominance. With this setup, you can see we're crossing a purple flowered pea plant versus a white flowered pea plant, and all of the offspring are purple in the F1 generation. So, a Punnett Square might look something like this. The purple plant could be homozygous dominant, so maybe we'll use, just for simplicity, R's. So if the purple is either heterozygous or homozygous dominant, it would have the purple characteristic. So let's do a homozygous dominant versus a homozygous recessive. So it gives us white flowers versus purple. And the allele combination is a traditional monohybrid cross. Set up so that all of the inside here would be purple. There is another option. You could also have another set of alleles being used to give us our purple color, of course. The purple color could be heterozygous, right? So we use the same setup, still be purple. And a white flower, right? And in this case, we can end up having recessive, which would be the white color. Now, we don't know if the three flowers are showing us all the possible outcomes or just the ones that were chosen. So either of these could actually be the correct response to the setup, because we just don't know. In regards, right, depending on whether the purple flower was homozygous dominant or heterozygous, we end up with all purple flowers eventually, right? So that's what we know. Most likely, the authors of this problem probably wanted us to think that it was homozygous dominant. So if the genotype was big R, little r, equaling 50%, and little r equals 50%, that would be the outcome if it were a hybrid versus a homozygous recessive. We also saw that it could be big R, little r hybrid at 100%, right? If the parent was a true, both homozygous but for opposite traits. In phenotype percent, we'd see, and the outcome here would be purple 50%, white 50%. And on the outcome here, they'd be at 100% purple if it was homozygous dominant versus homozygous recessive. Now, this is what we consider to be complete dominance. We know that if you have a dominant trait, it's going to overpower a recessive, right? So in this situation, we see that here, right? We have one dominant, at least in this situation where we have the hybrid, or if it was truly a homozygous dominant versus a homozygous recessive, right? They'd have both alleles. Anyways, we create the situation where you'd have the white skipping a generation if it were in fact creating all hybrids. Now let's compare that to the Snapdragons. One of the first things to notice on the problem here with the Snapdragons is we have a red flower on the Snapdragons being crossed with a white flower, and interesting enough, we get all pink. So some type of intermediate between red and white is created. So let's take a look at a Punnett Square with this. Alright, so we have red versus white. So how do we get the pink? Well, let's take a look with our Punnett Square. Alright, so we have a red versus a white. Now, this is going to show a new pattern of heredity called incomplete dominance. So we're going to create some alleles here that are going to show that neither one is dominant. So we're going to show that the red is homozygous for one allele. We'll use capital R, and we'll use capital W for white. Now, traditionally, when they show incomplete dominance, you may see them use something like R with a prime on it and R with a prime on it, just to show that. So let's plug it into the Punnett Square here. We're going to have a red flower versus a white. Right, so we've got R's for the red and W's for the white. Of course, we'll have the alleles RW throughout the Punnett Square. And what we get for a phenotype are the intermediates. They're all pink. Now, if you took two of these from the F1 generation, took an RW times an RW, you would end up with these heterozygous Snapdragons producing 25% that are red, 25% that are white, and then 50% that are pink. Because you can take those, put them back through a cross in a Punnett Square, and you can see that the parent colors come back out, as if they were to, again, skip a generation like we see. So the pink, right, is going to be seen for the F1 generation, F2 generation. You get back the reds and the whites, as well as 50% pink. We can show that in a Punnett Square. Alright, so we're going to take our incomplete dominant, and we're going to cross the F1 generation to make it F2. So we've got an RW versus an RW. Again, this is going to be a pink flowered Snapdragon versus a pink flowered Snapdragon. In this box, we get red Snapdragon, so 25% red. The lower right-hand side, WW, gives us a white. And then RW in two of the boxes gives us pink. So we sort of see this genotype ratio of 1, 2 to 1. So we get 1, 2 to 1, which would be 1 red for 2 pink for 1 white. So this pattern that we're seeing with this, when you get an intermediate, it's going to be incomplete dominance.